I will, as Eugenio said, I started my lab at Scripps Research uh, around 3 years ago. And the work that I'll talk about today was work that I started in Sasha's lab when I was a postdoc, and then took a little bit to get published. So uh, some of it was finished here at Scripps. So and I'll introduce like the people that did it with me along the way. But just to, and I will try my best to, I might skip over some slides because I think I maybe was a little overzealous in putting too much of the work in there. But nonetheless, I'll try my best to keep in time and discuss what we did here. So just to get us all on the same page, I always like to start by talking about what barrier tissues are. So our barrier tissues are the tissues that interface with the outside. And that means your skin, your respiratory and intestinal mucosa. And these tissues are very important for your health. They're our first line of defense with, again, the environment, and they have to defend us to this wide range of threats, but they're also exposed to a wide range of stimuli that are not bad for us. And in fact, some of this is good for us. And because of this, they have to exchange with the environment things like oxygen and nutrients. So because there's this constant exchange with the outside, barrier tissues have to, or they require this constant monitoring to make sure that there's maintenance of integrity of the barrier and that also pathogens are not coming in. And of course, we have to be able to discern what's an insult versus an innocuous agent. And today I'll be focusing particularly on the skin. Okay. Can you see my laser point? I hope you can see my laser point. Okay. All right. So yeah, so today I'll be focusing on the skin, which again is one of our largest interfaces with the outside. And again, it's exposed to all these different type of stimuli that some of it is good for you and some of it is not. And again, we have this constant need for patrolling for maintenance of integrity of the barrier and pathogens. And for this, we have well-positioned immune cells that live in the tissue. And these cells like sense and can sense when there's damage and pathogens through pattern recognition receptors. And when these get activated, they induce effector programs that start immunity and induce cell recruitment and mount a protective response. And in addition to immune cells, we also have a second layer of cells that can sense when there's damage or sense innocuous as well as non-innocuous agents that also are rich in the skin, which are sensory neurons. And these neurons induce, again, very much like immune cell, this protective responses for us in the shape of protective behaviors, things like pain, avoidance, like even itch for dislodging like let's say parasites or things like that. And so because of this, both the immune side and the sensory side both have to work to defend us and also maintain tolerance to things that are innocuous. And it's perhaps not surprising that there's crosstalk beyond just like inducing these protective responses independently. There's also crosstalk between them to make sure that this defense and tolerance balance is maintained. So we know that and much work has been done in the past 10, 15 years, looking at how immune cell derived factors, things like cytokines, like can induce the activation and sensitization of sensory neurons, inducing increased pain and itch. And this is a protective response to promote healing and preventing further damage on the site. or at least that's what we believe evolutionarily this has come from. But the reverse is also true, where factors made by sensory neurons, things like neuropeptides, can induce the activation and recruitment of immune cells and be involved in the mounting of an immune response. And so there's like lots of work to be done here. There's many factors we don't know, and this is a wide open field with many people interested, because it is like so likely so important and understudied at this point. So with this in mind, this is one of the things that I was interested in when I was starting my postdoc. And I went to the lab of Sasha Rudensky at Memorial Sloan Kettering. And I was really interested and Sasha's lab has been a pioneer and worked in regulatory T cell biology. And so when I joined the lab, I was really interested in how regulatory T cells maybe interact with neurons and maybe promoted this balance and tolerance to immune stimuli through a mechanism that involved neurons or sensory neurons. And so the work that I'll talk about today was, again, work that I started there. And I had these people here on the top that helped me, Paolo and Regina and Emma, and also Stas, who were at the time students in Sasha's lab, and then two postdocs in my lab who helped me finish the work once I was here at Scripps. And the work is, in case you want to look up, again, I will try my best to like not bore you with all the details on the paper, but if you are interested in looking at it more carefully, it has been published last year. So okay, so the reason why I was interested in and how regulatory T cells and sensory neurons may interact to promote this balance between tolerance and immunity was because, of course, regulatory T cells are an important or a key factor or key cell type in immune suppression. These cells are a specialized T cell lineage that is important for preventing autoimmunity and constrain inflammation. And we've heard, I think, all about them because of this year or last year's Nobel Prize. So I won't go too much in the background of them. But beyond their role in immunity, we know that they have other functions like outside of, again, immune regulation, that suggested to me that maybe there could be this other non-immune routes that they could take for mediating immunity or mediating other aspects of skin biology that we were not privy to. So, and the reason, one of the big reasons I thought that this could be the case was because early on when I started doing some imaging of where regulatory T cells were located in the skin, we found this striking co-localization with axons innervating the skin. So here, if you see, and the neurons here are labeled in white by tubulin beta 3, Treg cells are in yellow, and the blue is CD31, which labels blood and lymphatic endothelial cells. And what we found was that this co-localization of Treg cells with axons innervating the skin was not just secondary to their localization near blood vessels, which we can also find them near blood vessels, but when we plotted the localization of Treg cells towards axon versus endothelial cells, we didn't see any correlation. So these seem to be two independent or cells localized independently from one another. And that made us think that, of course, co-localization doesn't mean interaction, but it means that cells were close enough to either do something to regulate the environment around sensory neurons or to directly interact with neurons through either secreted factors or, in some cases, direct contact. to mediate some form of crosstalk. So we were interested. This really opened the door to there could be some interaction here. So I thought we thought, and Sasha thought too, that it was worth looking into this. So to functionally test this, we used mice that Sasha's lab had generated many years ago that harbor a diphtheria toxin receptor in the FOXP3 locus. So with these mice, you can, if you inject DT, diphtheria toxin, this induces the killing of cells that are expressing DTR. In this case, is FOXP3. And I will say that this has been a model used by many people at this point, and it's like very nice and very clean. And because it is very effective, it induces really rapid onset of autoimmunity. And so we wanted to make sure that whatever effects we were looking at in terms of its effects on sensory neurons were not just downstream of widespread inflammation, which does occur quite fast when you deplete regulatory T cells, again, because these cells are very important. And so what we tried was 18 hours after treating FOXP3 DTR mice with diphtheria toxin, and then see what effects we had on behavior. And so at this time, we saw that the cells were like kind of gone. For sure, in the lymph nodes, they were depleted. But in the skin, we could still find some Treg cells, so we didn't see a significant change in their numbers, but these cells, whatever cells are there, are poisoned by the DT because they're not functional. So if we look at, for example, IL-10 production by them, after restimulating them, we see that the cells that came from mice that were treated with diphtheria toxin are not functional. So this gave us a nice window where like, okay, we don't see any signs of widespread inflammation because we don't see changes in other T cell subsets. We didn't see any changes in any myeloid cell subsets. We didn't see any changes in cytokines, neither in the lymph nodes or the skin. So at this point, it was like we had gotten rid or poisoned the cells, but hadn't had like inflammation kick in yet. Okay, so with this setup, we looked at how the mice, how sensory perception was changed in mice. And we found that mice had an increased sensitivity to heat stimuli. So if we put mice in a hot plate, that it's an uncomfortable temperature for them, they will do this we call them nociceptive behaviors, where they will lick their paws to try to cool them, or like jump, so take their hind paws like off the plate. And we saw that the mice have a shorter response to heat. We also saw that the opposite is, we test the opposite for cold, where we put acetone, which feels very cold in the paw. And then we see how long the mice are lick it to try to get rid of that cold stimuli. And we see that when mice were treated with DT was longer, so they sense cold much more strongly. And then this was also true for mechanical stimulation, where we give different filaments in the paw and then see how often the mice retract every time you test with different strengths. And what you can see here is that when we treat mice with diphtheria toxin, or when we deplete regulatory T cells for this short amount of time, that the mice have increased sensitivity to the thinner filaments compared to PBS treated controls. So this again suggested that this increased sensitivity was across multiple types of stimuli. We also wanted to see if this was true for more chemical type stimuli. So we wanted to try capsaicin, which is the component in hot pepper. So what we did was, and if you've ever put something that has like a lot of, again, some like very spicy sauce, and you get it on your skin, you can actually feel the heat on your skin. So that's kind of what we did to the mice. We put a solution of capsaicin in their ear and then tracked how long, and after 30 minutes, we tracked how much activation we could see in the in the neurons that innervate the skin. And so just very quickly, I will tell you how we did this. So the neurons innervating the skin are their nuclei are far away from where the axons are. So the nuclei are housed in the structures called the dorsal root ganglia, which you can see here. And they extend axons all the way, and this happens all throughout the body to innervate different areas. But for the skin, we did some mapping and saw that and us and others, I should say, did some mapping to trace ear innervating sensory neurons, and we found that they are located in the cervical dorsal root ganglia, which are located in the upper part of the spine. And these guys connect they have bifurcating axon. One axon goes to the tissue, so in this case, the skin, and the other one goes to the spinal cord, which then transmits information to the CNS, so transmitting information to the CNS. Okay, so in these experiments, what we did was we, again, depleted regulatory T cells using FOXP3 DTR mice, treating them with DT for 18 hours. And then after that, we put capsaicin on the ear of the mice. And then we waited for 30 minutes and then looked at how much activation there was in the dorsal root ganglia using a staining for FOS, which is a rapidly induced transcription factor. This happens in neurons. It also happens in T cells also express FOS, which is something that often I think neuroscientists are surprised to know. But these are just like T cells, neurons get rapidly activated, and they do transmit information much faster than T cells do, but they all upregulate FOS. So in this case, we were looking at DRG, so the cervical DRGs, and we're staining for FOS, and we did it in a way that we gated on tubulin beta 3 positive cells, so neurons in the dorsal root ganglia, to make sure that we were looking at neuronal FOS induction. And what we saw was that we can see nice induction so if you see the first and the third column, we see this nice induction of FOS in the DRG with when we give the mice capsaicin in the skin. And this induction was much more pronounced in mice where regulatory T cells were depleted. Therefore, that suggested that we were seeing more sensory neuron activation when Treg cells were not there, very similar to what we saw in the behavioral assays. And so the question then was like, okay, so it seems like regulatory T cells can modulate sensory neuron activation. What does this have to do anything with inflammation, or is this a completely separate, these two independent mechanisms? And so in order to test this, we wanted to look at an inflammation model that we know is neuron dependent, because we know that sensory neurons, as I told you in the beginning, are able to induce the activation and recruitment of cells for some immune responses. And this psoriasis-like disease is a good example of this. So this has been well-established. Work from Uli von Andrian lab really showed this very nicely. And also where they show that if you get rid of sensory neurons, mice don't develop psoriasis in response to imiquimod. But the other kind of the flip to that, which is also super interesting in a very nice paper, is that when you activate these neurons independently of giving them any imiquimod or any other stimuli, just with in this case that we're using photoactivation, just the activation of sensory neurons in the skin is enough to induce this type 7 response, and this is very psoriasis-like disease that is induced in response to neuronal activation. So in our case, because we are seeing this increased neuronal activation with regulatory T cells being gone for a short amount of time, the prediction would be that this would enhance or make disease worse, like enhance inflammation. And so we tested this by basically doing a version of the experiment that I mentioned before, where we gave mice imiquimod. Again, we apply this in the skin to induce type 17 response in the skin. And for the last 2 days, we depleted. And so the initiation of disease happens and by day 3, you start seeing IL-23 production by dendritic cells, which then induces IL-17 production by dermal gamma delta T cells. And so what we're looking at the point where IL-23 is going up. So on the third, after 3 days, we collected tissues and looked at one in the dorsal root ganglia where was there any more activation of sensory neurons? And we do indeed see that if you deplete regulatory T cells, you see increased activation compared to the controls in imiquimod-treated mice. And this translated to, as you would predict, more IL-23 production in the skin. And we saw that this effect was gone if we remove the neurons chemically, but I won't go into too much of that now because of time. So, okay, so knowing this, we're interested on how can this happen. So we know that regulatory T cells can affect neuronal or sensory neuron activation, and this seems to have an effect on inflammation. Is this a direct effect or is this through something else? And we did this scouring of basically taking gene expression data, and Sasha's lab has basically sequenced regulatory T cells in every shape and form. So we had a lot of data to go through. And what we did was look at what we started with neuropeptides because, of course, we wanted molecules that maybe immune cells could make that could communicate with neurons. And we found out this really interesting candidate with this gene called PENK, which was highly enriched in regulatory T cells and not other conventional T cells. And the reason why this was so enticing was that PENK PENK is a gene name for proenkephalin, is a precursor, and proenkephalin is a precursor for enkephalins. Now, these are these endogenous neuropeptides that inhibit pain perception, and they do this by binding opioid receptors. And yes, these are opioid receptors, which are the same thing that bind opioids, which are exogenous substances that decrease pain. So your body has the same version or the natural version of these peptides that bind these receptors, and they decrease the activation of neurons that transduce the sensation of pain. Very interestingly, when we did some fate mapping using PENK-RE, a PENK-RE reporter, we found that regulatory T cells that expressed or had a history of PENK expression were very enriched in the skin. So this seemed like a reasonable candidate. And I won't go through all the data, but we did see that if we got rid of regulatory T cells that expressed PENK, we saw this increased activation of the neurons and increased inflammation. And if we did the opposite, if we got rid of all Treg cells except the ones that express PENK, we saw that this was gone, that this effect was gone. So this suggested that this subset of regulatory T cells that expressed PENK were the ones that were inducing this sensory neuromodulation. So, of course, if PENK itself is doing this, you would want to know if regulatory T cells need to express PENK to decrease sensory perception and decrease inflammation in this way. And so we deleted PENK using a PENK flox mouse with a FOXP3 CRE-ER, which is expressed in regulatory T cells. And what we saw was that if we deleted even long-term deletion of PENK and regulatory T cells didn't induce in and of itself and I'm very short on time, so I'll be very fast. But it didn't induce in itself an increase in inflammatory state. We did see that it induced this increased sensitivity, both in the sensitivity to heat and mechanical stimuli, but very interestingly, not too cold. So this seems to be a completely different mechanism employed by Treg cells that's PENK independent. And very quickly, I will tell you that that these cells also regulate this type 17 inflammation in the skin. PENK in regulatory T cells is required for dampening this inflammatory state in the skin. This is neuron dependent. Again, I'll be kind of fast and I'll go to the punchline, which is if you delete this neuropeptide in Treg cells, you induce psoriasis in these mice, the mice get sicker. So we see increased 17 production in dermal gamma delta T cells. We see more neutrophils coming in, and we see this thickening of the skin because of the inflammation. And we found, again, the paper is out, so you can see all these details. We found that this is dependent on glucocorticoid receptor signaling and T cell receptor signaling. But maybe most interesting of all, we were really interested in what neurons Treg cells have to talk to induce this effect. And to do this, we did some single cell sequencing of sensory neurons to see what neurons were changing. And this was done by a postdoc in my lab, and Stas, who was at the time in Sasha's lab, and he's now at Scripps also. And we found that there's this subset. So many sensory neuron subsets had changes in gene expression, but the ones that the ones that had the most interesting set of gene expression changes were these neurons that express MRGPRD, which are this polymodal sensory neurons. And the reason why we thought they were super interesting is because they had this program that has been correlated with inflammation. So these genes that I highlighted here that are upregulated have been associated with increased inflammation, including the gene for CGRP, which has been linked to this induction of type 17 immunity. So this is the model that we have now, where a noxious stimuli will induce production of proenkephalin from Treg cells, which then gets processed into enkephalins, then bind opioid receptors and MRGPRD neurons to decrease their activation and their sensitivity to promote or to prevent this pro-inflammatory program. And the lab is really quite interested in following up here. The lab is really interested in following up what the role of these endogenous opioids is in skin and other behaviors and this neuromodulatory connection. But we're also interested in how other subsets of neurons regulate immunity and how they sense different type of immune responses. So that's what the lab is doing now in this space. And with that, I am so sorry. I think I really tried to put way too much into the presentation. But these are I just wanted to acknowledge very quickly the people who did the work. So I mentioned, so this is my lab, and these are the two postdocs in my lab who helped with the work that I talked about. Again, all the work was for the most part done in the Rudensky lab, and these are the people in Sasha's lab that helped with the work. And then Andreas Zimmer generated the PENK flox mice, and they were very generous in giving us some sperm a few years ago so that we could reconstitute this line. And we also had some help from the behavior core research, Scripps research, and from the DAIA lab at Weill Cornell for some of the behavior experiments that I talked about. And these are the funding sources that have supported the work. And with that, I will, I'm sorry that I went a bit over my time, but I will stop here. And thank you.
Thank you so much, Alejandra. So I guess we have time for the 2 or 3 questions. If anyone, you can type in the chat, or you can open your mic. I guess first of all, I will congratulate you. I will ask you if I think